This video is more of kind of an evolutionary and developmental biology video that's trying to just assess this question, which is signal molecules have a function within the body, but certain regions of the body can use the same signal molecule, but they have different abrupt functions. So like the function of GLP-1 in the gut is different from GLP-1 in the brain, but the semantic information that's conveyed by that molecule is generally the same. So for example, the signal molecule GLP-1 within the gut is used to convey information about the nutrient status within the intestines. So as food moves down the intestines, imagine this is our little gut tube right here. This is our gut tube, and I have cells that line the small intestine called L cells. They're detecting the presence of nutrients here and releasing GLP-1 into circulation, GLP-1. And GLP-1 is an incretin hormone, so it's acting on the pancreatic beta cells, improving their ability to synthesize and secrete insulin. And GLP-1 is acting on G protein link receptors, causing an increase in cyclic AMP. So this is all happening within the body, usually it's in circulation, this is in circulation, but it has a hard time crossing the blood-brain barrier. Whereas there's also GLP-1 in the central nervous system that's spatially distinct from the GLP-1 within the gut, but it has a similar contextual effect where it's signaling the organism about food is available. Now is not necessarily a time to consume, but it's a time to be satiated and focus on other things. So this is an example of how the same signal molecule can be used in different tissues for different functions, but overall it has the same contextual effect, or it has the same semantic meaning. So right here, this idea that the signal molecule doesn't necessarily equal function. What equals function is the signal molecule binding to the receptor it's intended to bind to, the downstream effects of that receptor on the cell, and then the contextual environment of the cell. So it depends on quite a bit of information. It's all contextual. The GLP-1 that's binding to GLP-1 receptors located on the pancreatic beta cells will still activate cyclic AMP pathways. So imagine this is our little G protein link receptor. It's spanning the membrane. It'll activate a G protein, which binds to adenylate cyclase, converts ATP into cyclic AMP. And then cyclic AMP is our second messenger in this case. And cyclic AMP will facilitate the secretion of insulin in the presence of glucose. So it's an incretin hormone. It's enhancing the effect of these pancreatic beta cells. And GLP-1 binding to GLP-1 receptors in the brain, turning cyclic ATP into cyclic AMP, that whole function is the same. So same receptor, same signal molecule, but the function within neurons is different. So it's acting in the brain as more of a neuromodulator, increasing the rate that some neurons are firing, like those POMC neurons, the pro-opiomelanocortin neurons. And so this is an example where the signal molecule doesn't necessarily equal the function. What matters is what the signal molecule is binding to, the downstream effects of that receptor, and what it causes the cell to do, but also the contextual environment of the cell. So there's quite a few examples of these hormones or these peptides that are released from the gut, so they're gut derived, but they're also present within the brain and they act as neuromodulators. So one example is that GLP-1. Another example is somatostatin or peptide YY or CCK or ghrelin. They're all expressed within the gut. They have effects on signaling the body of an organism to react to the presence of nutrients, but they're also present within the brain. And so this is kind of more of a diagrammatic example of how this works. This is supposed to indicate the gut tube right here, so the small intestine, and where the presence of nutrients, in this case amino acids and fats, will trigger the endocrine cells of the small intestine, so cells that are producing and releasing a hormone into circulation of the organism in response to nutrients in the gut. In this case, they're releasing CCK, or the cholecystokinin. And CCK will travel in circulation and have effects directly on cells, but it's also present independently within the brain. Same as GIP, it's present independently as a neuromodulator within the brain. The GIP released from these GIP releasing cells within the gut is spatially distinct from that within the brain. And this kind of goes to this broad idea that in early organisms, these peptides were sensing nutrients, and they would have been able to convey signals to tissues throughout the body, including the brain or the central ganglia. But in us, we have more of a vertebrate-like blood-brain barrier that spatially separates the body from the brain, but the contextual meaning of GLP-1 remains the same because it was inherited from earlier metazoans or animals that lacked that spatial separation. So these peptides are ancient. They evolved before a vertebrate-style blood-brain barrier. So if I had GIP being released from these cells within the gut that are sensing nutrients in an early organism, that GIP is going everywhere. Whereas now it's still going throughout circulation, but it has a hard time crossing the blood-brain barrier because it's a peptide hormone. Some can and some can't. I'm not entirely sure if GIP has a great ability to cross the blood-brain barrier. I know that GLP-1 doesn't cross the blood-brain barrier that well unless the concentrations of the substrate are really high. So in the case of taking a GLP-1 agonist, you're raising the overall concentrations of that molecule beyond physiological levels. So they're getting into the supraphysiological level, almost upwards of 1,000 times what is normally released in the presence of nutrients. And that allows that GLP-1 to kind of oversaturate the blood-brain barrier and allow it to move across that barrier and influence the brain as a neuromodulator. And so in organisms like us with a vertebrate-style blood-brain barrier, these peptides evolved before we had that. So they're having effects in the brain as neuromodulators, but also in the body as hormones. And so even with organisms with a blood-brain barrier, there's interoceptive information like the vagus nerve or circumventricular organs in the brain that can relay information about the body to the brain. So even though this GLP-1 may not be able to cross the blood-brain barrier, there's regions of the brain that are exposed, those circumventricular organs, or the vagus nerve, the sensory endings that are present within the gut that relay visceral, afferent sensory information from the gut to the brain that have GLP-1 receptors that can tell the brain about the contextual environment of the body and the hormonal environment after sensing nutrients. So it's plausible to think of a scenario where GLP-1 acts as a nutrient sensor in the gut and signals to the body that nutrients are available, but then has the opposite effect in the brain, like stimulating hunger. And this is just a hypothetical, but it isn't really evolutionarily consistent with the general semantic meaning of a regulatory peptide because those molecules evolved prior to the spatial separation between body and brain. And there really isn't much of a spatial separation. There's so much interoceptive integration where you're relaying information to the brain about the body that it doesn't really exist. So there's the same molecule like GLP-1 will have an effect on the body that's consistent with nutrients are present, but it also has an effect on the brain produced by neurons within the brain as a neuromodulator that has the exact same effect that nutrients are present, act accordingly. And that's the same with something like cortisol, where generally it's seen as a hormone that's involved in adaptation to stress throughout the body. And that could be metabolic stress, it could be psychosomatic stress, it could be immunological stress. So it varies, but overall it has a semantic meaning. And that doesn't mean that it's just a molecule that binds to the receptor, has downstream effects, It's contextually different in different cells, but overall has a general function towards signaling stress or signaling that we have nutrients or we don't have nutrients. And so the gut and the brain overall just use a common molecular language that was inherited from earlier metazoans regarding what these molecules mean. And so this is kind of an interesting example where there's this gene called the proglucagon gene that's present within the cells in the gut that produce glucagon or GLP-1 or GLP-2. But it's also like the same gene is present in cells throughout the body, but different cells have transcription factors that can bind to enhancers that promote the transcription of this gene, and that the transcription of this gene can also occur in the brain. So like right here, like this brown region, this blue region, and this black region are all what are considered enhancer regions. So they're non-coding regions of DNA that are influenced by proteins, and these proteins can come over and bind. Let's imagine this blue guy could come over and bind this region, and this region can fold over and help recruit an RNA polymerase to this promoter region to facilitate the transcription of this gene into mRNA right here. So this is my little mRNA strand, which can then be translated into a functional protein called proglucagon. And so I can create this proglucagon in multiple different tissues depending on the transcriptional environment. So even though the transcriptional environment in the enteroendocrine cells in the gut is different from the cells, the proglucagon releasing cells within the brain that release GLP-1 as a neuromodulator, I can still produce this protein called proglucagon. And then each of these cells can have a different transcriptional environment that codes for proteins that can regulate what this proglucagon is eventually cleaved into. And so there's little proteases that are present in, let's say, the enteroendocrine cells or the brain or other cells in the body that determine this proglucagon will be cleaved into glucagon, or it'll be cleaved into GLP-1 or GLP-2 or these two molecules, which I'm not that familiar with. But overall, there's levels of regulation in terms of what these molecules do in tissues, where they come from, the transcriptional environment that elicits these molecules being present in different tissues. But overall, these molecules have a general semantic meaning, and they relay information to